next speaker, Dr. Flavio Frohlich. Flavio.
Good morning, everyone. Hamed and team, thank you so much and congratulations on organizing this wonderful webinar. It's a pleasure to be with you all here today. Next slide, please. Here are my disclosures. You can also find them on my website. Next slide, please. And before we dive in, I just want to make sure to share with you that the work I'll be showing you is not my work, but it's the work of the amazing team I have the privilege to lead. Next slide, please. So, using electric fields to modulate brain activity, I think in some sense it's humbling to remember that it's not something we've invented, but rather there's quite convincing evidence at this point that the electric fields that are strong enough for us to measure with EEG electrodes, great convenience clinically and for research, but that's not the main purpose. Rather, these electric fields that emerge from orchestrated, synchronized, rhythmic brain activity actually directly influence neuronal activity and help to solidify and strengthen emerging and ongoing oscillatory patterns. So in many ways, what we're doing here is copying what evolution has developed as a main mechanism for organizing neuronal communications beyond the classical synaptic transmission. Next slide, please. And we can replicate and study this in a reduced animal preparation. And you've already heard from previous speakers about the amazing work done across the globe at this point, looking at how weak rhythmic electric fields seem to be particularly effective at modulating neuronal activity, as shown here in a reduced animal preparation of an oscillating network that entrains or locks in or synchronizes to the applied perturbation. It's really important to keep in mind that at these low amplitudes, the goal is, as we've heard, not necessarily to drive neural activity to turn on and off the switch. The brain is much more complicated in terms of endogenous dynamics, and primarily what we know now is that these weak rhythmic perturbations actually alter the phase or the timing when neuronal activity occurs. Next slide, please. And we can see that when we track the occurrence of each bout of neuronal activity as part of a rhythmic overall activity pattern and track that over time and demonstrate that we apply these weak rhythmic perturbations, we're not changing the activity level, but rather we change the timing and the structure of when this neuronal activity occurs by adjusting the phase. So if you're asking what is the dose and how is the response in terms of number of neuronal spikes, which is still, I think, a very prevalent model, at least conceptually, that many of the community are going after, we might be missing what we have to look for when we use low amplitude perturbations. Next slide, please. So I just want to share with you that I think before we do animal studies, there are computational simulations, and we've already in previous talks this morning heard about the great advances in terms of modeling the delivery of electromagnetic energy to neural tissue. And we've also heard that the response to the waveform and structure of those electromagnetic perturbations really depend on the state of the brain. And that is an exciting question, but it's also a parameter space explosion of kind of hard to fathom dimensions, because now we're not only facing the unlimited choice of different parameters defining our waveform, but also we have to deal with the rich underlying dynamics that change from moment to moment, that change from person to person, that probably also change from species to species to understand what the stimulation does. So computer simulations allow us kind of rapidly, despite the oversimplification any computational model necessarily is, to get a more parameterized, more comprehensive understanding of how the response to stimulation depends on brain state. So this is just kind of an introductory illustration of a computer simulation demonstrating this entrainment effect that I've just shown you in this reduced in vitro preparation. Next slide, please. And what we can do then is, in contrast perhaps to animal experiments, we can build networks that have different types of activity patterns. In this case, we're looking at a network biophysical simulation was built to exhibit a 3 Hertz oscillation, and now we can systematically probe the different stimulation parameters, at least in this case the amplitude and the stimulation frequency, to understand and predict the response to stimulation. And what we see here is that the response to stimulation depends on the tuning of the stimulation waveform to the endogenous dynamics. So when the stimulation waveform was aligned and tuned to the ongoing endogenous frequency, illustrated here in hot colors, we had an enhancement and entrainment of the neural activity. However, if there was a detuning or difference between the stimulation frequency and the ongoing simulated activity, the stimulation could actually have the opposite effect. And this overall structure here is referred to as the Arnold tongue, and has been previously suggested and studied actually centuries ago in terms of how mechanical systems such as watches and metronomes and clocks respond to rhythmic periodic perturbations. So there is an underlying dynamical systems and theoretical physics kind of modeling and thinking approach we can use to understand this. But the key point there really, I believe, is that if we don't measure brain state, if we don't know if someone what the specific frequency is, and we just apply a given waveform and expect that whatever frequency we put in, that's what we're getting out, we're really kidding ourselves, because as you can see here already in this oversimplified simulation, you can really actually get the opposite effect for these detuned waveforms, such a suppression of the ongoing oscillation, further illustrating the importance, what we've already heard in these wonderful introductory talks we had today about the importance of measuring brain state. Next slide, please. And just another point to the variability and how low we go question. So here's a computer simulation again, where we varied the dose by changing number of neurons responding to the applied electric field. And what we found is that for these low amplitude stimulation, there's highly variable outcome. This is from simulation to simulation. So it's still structure same network, but just with a small change in overall parameter setup. And once we increase the amplitude past this roughly 50% of neurons being targeted threshold, then there was a robust and reliable response to the stimulation. Quite likely in most of our studies, we're actually at the lower end of this distribution. So realizing that again, the even small differences in endogenous dynamics greatly shape the response to stimulation. Next slide, please. And then further on, as we have richer state dynamics, as we observe in the intact brain, where we have these dynamic changes between different spatiotemporal activity patterns, as shown in these computer simulations of two interconnected networks, the same question gets even more acute, because as the endogenous dynamic change, we got to think about how ideal in real time adjust the stimulation to maintain that tuning and successful target engagement through the applied stimulation, something that still the vast majority of stimulation studies, of course, do not do, because there's a predefined locked-in stimulation waveform that most, every study participant would receive. Next slide, please. And in fact, even just at the level of two connected networks, still a very reduced simple system, we start to see that stimulation, in this case, in-phase synchronized stimulation, can have the kind of perhaps naively predicted and hoped for effect of synchronizing the two networks shown here to the left. But it also, same network, just slightly different initial condition, meaning slightly different state. As we're applying the stimulation, the effect is actually opposite. It can break the synchronization. And the effect of the stimulation beyond the applied waveform when we turn off the stimulation also depends on the endogenous state. In this case, you see how the oscillation was disrupted and maintained disrupted and did not find back to its previous synchronized state after conclusion of the stimulation assimilation. Next slide, please. Now, we can, as we start to have these understandings, we can go and test them in animal preparation. So here's an in vitro preparation where we, with optogenetic stimulation, use the rhythmic pattern, and we induce a strong rhythm. And the takeaway message from this is essentially that whatever electric field frequency we applied for these low amplitude perturbations, still the dominant rhythm was the endogenous generated rhythm, indicating again this non-linearity we heard about as a function of strength of oscillation. Next slide, please. And what we then see here to the left and to the right is for two different stimulation amplitudes in vitro, that for lower stimulation amplitudes at 1 Hertz, the yellow line, where we match the stimulation to that endogenous-like oscillation, we had the stronger enhancement. However, on the plot to the right, when we cranked up the electric field to a few millivolts volts per meter more, then that frequency tuning did not matter anymore. All the stimulation amplitude has a similar effect and entrained the network. Next slide, please. Now, these are all reduced in vitro preparations, and the question is, how general is the principle? I don't have time to talk about this in detail, but these entrainment principles of the Arnold tongue appear to hold for different simulation modalities and for different networks, as shown here for thalamic networks simulated with more DBS-like, pulse-like stimulation. Next slide, please. And then finally, due to translation animal studies, these principles heard about, do they actually apply in animals? And again, there's a question of choice of model species, because we want to make sure, not only thinking about brain size and geometry we heard about, but we also want to make sure we choose as an animal species that endogenously exhibit the kind of rhythm that we want to target in human studies. And this is one of the reasons why I've been investing a lot of our energy in developing the ferret as a model species, as it exhibits, as you can see in this slide here, this endogenous alpha rhythm that we're so interested in human studies that rodents such as mice and rats do not exhibit. Next slide, please. I'm not going to talk about this because we have the great privilege of having Dr. Peter Jeff here. This was a wonderful collaboration. So he will talk about the electric field simulation. I want to thank him for the continued and great collaboration over the years. Next slide, please. And maybe just very briefly to wrap up here. Here I'm showing you the effect as a function of the tuning on the x-axis, a function of simulation amplitude on the y-axis, the response of two presumed cell types to low amplitude TACS. And what you can see here on top is that we again find this inverted triangle demonstrating the presence of the Arnold tongue, not only in this computer simulation, but also in the intact animal. And on bottom in purple, you see no structure because that is measurement change in firing rate, demonstrating there's no change in firing rate, again highlighting that low amplitude periodic perturbations do enhance network structure but not overall activity level. Next slide, please. And again, we can then loop back to computer simulations. I know this morning is about animal work, but I just want to illustrate and remind the audience and also newcomers to the field that we can really leverage computer simulations to get even more mechanistic insights and also minimize the number of animal experiments that need to be done. And this is just a brief pointer. It's published so that we're able to replicate some of these experimental findings in a biophysical simulation. Next slide, please. Now, with all this, I just want to, this is my last slide, I promise, just want to zoom out. And, as we're starting to understand the mechanism better, that's reassuring, and we can kind of maybe put behind us some of these more simplistic questions. And I really appreciate a good colleague and friend Marom. I'm also offering for us to move on beyond, like, does it work, does it not work, how low can we go? Because ultimately, I think with a community we can accomplish, as we better understand the mechanism of action, we can start to rapidly translate these in neuroscience informed mechanism-driven, using rational design pipelines to develop novel interventions to target pathological oscillations to treat hard-to-treat conditions, such as in this case here, shown chronic pain or depression or other applications where these low amplitude periodic perturbations start to look really promising in randomized controlled trials. So just to make sure we keep the big picture in mind, I want to again thank you all for your presence and the organizers and the other speakers for putting this wonderful event together. Thank you very much.
Thank you very much, Flavio.